Welcome to the second part of the digestive system. In the first part we saw that how the food reaches to the stomach, and now we will see that what happens further than that. Now see, when the food reaches to the stomach, it is mixed properly with the gastric juices. Now it is called chyme, and this chyme exits the stomach through pyloric sphincter, and now it enters into duodenum. Now duodenum is the C-shaped organ or the C-shaped part of the small intestine. Now when the chyme enters the duodenum, it is thoroughly mixed with Brunner's gland secretion, the bile juice and the pancreatic juice. All these juices are alkaline in nature. They neutralize the acidity of the chyme, and now this alkaline food is called chyle. Now upon the entry of the chyme into the duodenum, the HCl present in the chyme stimulates the mucous membrane to secrete some enzymes. Now these enzymes are hepatoquinone, cholecystokinin, secretin, pancreozymin, enterogastron and enterocrinin. Now we will see what are their functions. Let's see. So first we have hepatoquinone. Hepatoquinone stimulates the liver to secrete bile juice. Now see, the bile juice is actually secreted or produced by the liver, and it is stored in this gallbladder. Now the second enzyme, which is called cholecystokinin, it contracts the gallbladder, so that when the gallbladder gets contracted, the bile juice flows through the bile duct and reaches to the duodenum. Now the third hormone, which is called secretin, stimulates the pancreas to produce pancreatic juice. Now the next hormone, which is called pancreozymin, stimulates the secretion of pancreatic juice into duodenum. The next one, enterogastron inhibits the secretion of gastric gland in the stomach because there is nothing right now. And last, the enterocrinin or enterocrinin, this stimulates the food into duodenum so that the food move in a fast manner and reaches to the jejunum. Now we will see, as we said, that the chyme is mixed with three kind of secretion, that is Brunner's gland secretion, bile juice and pancreatic juice. So we will talk about them. So let's first talk about this Brunner's gland and its secretion. Now first of all, it is very important for us to know what are the Brunner's glands. Now see, the Brunner's glands are compound tubular submucosal gland. You must be remembering that, as we had seen, that when we talk about alimentary canal, it is made of 4 layers. That is, the outermost layer is called serosa, the second one is called muscularis, then it is called the submucosa, and the last one is called the mucosa, which is the innermost layer. So these glands, Brunner's glands, these are situated in the submucosa. That is why they are called submucosal glands, and they are group of glands. That is why they are compound glands. They are tubular. They are like tubes. So because they are compound, they are called compound. Because they are tube-like, it is called tubular. Because they are located in the submucous or submucosal layer, that is why they are called submucosal glands. So in this way, we can define Brunner's gland as compound tubular submucosal glands. Now what is the main function of these glands? Okay, so actually these glands are found in duodenum, so they are also called duodenal gland. And what is the main function? The main function of these glands is to produce a mucus rich alkaline secretion. Now you have to remember, mucus rich alkaline secretion, it contains carbonates. Now what are the purpose of this kind of secretion? So first purpose is to protect the duodenum from the acidic content of the chyme. Second purpose is to produce or to provide an alkaline condition for the digestive juices or the intestinal juices to be active. And the third important task or the function which the Brunner gland secretion performs is the lubrication of duodenum. Okay, so these are the three functions. First, to protect the duodenum. Second, to provide an alkaline condition. And the third one is the lubrication. So these are the major function of the renal gland secretion. After this, we will see the bile juice. Now, as I have mentioned that bile juice is produced by the liver and it is stored in gallbladder, from where through bile duct it is secreted into the duodenum. Now see, when we see, these are the parenchyma cells of the liver which produce the bile juice, and when we see the bile juice, it contains three organic substances. The first one is bile salt, the second one is bile pigments, and the third one is cholesterol. Now see, the bile salts are inorganic salts. They emulsify the fat particles and they help in absorption of fat soluble vitamins. That means vitamin A, D, E and K. So A, D, E and K, these are the four vitamins which are fat soluble, and this bile salt help in absorption of those vitamins. Now the next one, as we said, that is bile pigment. So there are two bile pigments. One is called bilirubin and another is called biliverdin. So bilirubin is reddish in color, biliverdin is greenish in color. Now the third thing is cholesterol. The third organic substance is cholesterol, which is produced by the liver. Now the cholesterol is an excretory product. It is not good for the body to have. After some certain amount, it produces a kind of burden which can cause many problems. So cholesterol is an excretory product which has to be expelled from the body. Now, so this is all about the bile juice. Now we will see what are the functions of bile juice. So the first function, which is very important, is that bile juice increases the peristaltic movement of the intestine. In this way, it helps in mixing of digestive juices with the food. The second important function is that the bile salts which are present in the bile juice, they make the food alkaline, and in this way they neutralize the acidic nature of chyme. First thing. Second thing, the pancreatic juices are only activated in alkaline medium, so it is very important for the food to become alkaline. So this is the second important function of the bile juice. The third important function of the bile juice is that it acts as an antibacterial agent. It destroys all the harmful bacteria which is present in the food or in the chyle. Now the next and the last one is the pancreatic juice. Now as we have seen that secretin is an enzyme which stimulates the pancreas to produce pancreatic juice. Now that pancreatic juice is released in the duodenum through pancreatic duct. Now there is another enzyme which stimulates this function, that is pancreozymin. So now see, pancreatic juice contains some factors in it, some elements in it. So we will see what are those. Now these are trypsinogen, chymotrypsinogen, carboxypeptidase, elastase, amylase, it is also called amylopsin, lipase, it is also called stepsin, and sucrase, maltase and lactase. So these are the group of enzymes which are actually released in the form of pancreatic juice. Now we will see what are their functions. So first of all, trypsinogen, it is not an active hormone, active enzyme. It is activated in trypsin by enterokinase. Now trypsin is the most effective proteolytic enzyme. Now I said the word proteolytic. What do you mean by proteolytic? Proteolytic, that means anything which breaks the complex structures of protein into simpler one. So those enzymes which break the complex structure of proteins into simpler one are called proteolytic enzymes. So the trypsin is one of the most effective proteolytic enzymes which act on all the proteins which are attached by pepsin. Now second one, chymotrypsinogen. Chymotrypsinogen is activated in chymotrypsin. Chymotrypsinogen is not an active enzyme. It is activated into chymotrypsin by trypsin itself. It is also a proteolytic enzyme. Now carboxypeptidase. Carboxypeptidase is activated by enterokinase and trypsin. It also performs the same function. Now next is elastase. Now elastase can convert or emulsify the elastin. Now the amylase is also called amylopsin. Now what is the function of this amylase? It converts the starch into maltose. Now here we have to understand that when I say starch, it is polysaccharide, and maltose is a little bit simpler form of the starch, or the little bit simpler form of sugar. Now next is lipase. Now see, lipase ends the digestion of fat by converting it into fatty acid and glycerol. So the enzyme which acts on fats is lipase. Lipase breaks down the complex fat particles into simpler one, which are called fatty acid and glycerol. The next one is sucrase. Now as the name suggests, okay, sucrase converts the sucrose. Sucrose is also a kind of sugar. So sucrase converts the sucrose into glucose and fructose, which are rather simpler form of sugar. Now the next one is maltase. As the name suggests, okay, it acts on maltose. We remember that amylase converts the starch into maltose. Now that maltose is acted upon by maltase, and this maltase converts the maltose into simple glucose. Now the last one is lactase. Now lactase, as the name suggests, it converts the lactose into glucose and galactose. Again, these are the simpler form of the sugar. So see, here we have to keep one thing in mind, that whatever we eat, first of all, it is broken down into simplest form and then it is absorbed. In case of sugar, the simplest form is glucose. That is why whenever you see any ad of glucose in the TV, it is always said that glucose instant energy, because it is not needed to digest it, or it is not needed to go through the complex process by which your food generally goes through. So in this video, in the third video, we will complete this chapter. Thank you.